Hi, I'm Sara Pacheco and I'm a student at Northern New Mexico College, and I am an intern in the Bioscience Division at LANL. And today I'm going to talk to you about our research converting algae into biofuels. And more specifically, I'm going to talk about a salinity tolerance study that we did on a genus of algae called chlorella. So a little about plant-based biofuels. These are biofuels that are produced from biomass as opposed to fossil fuels. And some common crops that are used for plant-based biofuel production is corn, soy, and hemp, which you can see here in the picture. Biofuels are very beneficial because they are carbon neutral, which means they use up as much carbon they use up as much CO2 as they grow that they will burn once when they're harvested and used for fuel. They're a renewable resource and they are easier to harvest than fossil fuels, and they feed into our current fuel systems, which means that we wouldn't have to make any changes to our current engines. There are some challenges with plant-based biofuels, such as they compete for land for our food source. Crops that are growing lots and lots of corn for biofuels could be used to grow our own food. And also it's an increased use of water, which is a non-renewable resource. So algae is an excellent source for biofuels because it has all of the benefits of plant-based biofuels and more. As you can see here in the figure, growing up algae can produce a lot of different things that can be beneficial, like plastics and fuels, food supplements. It can be grown in places where organisms can't live due to extreme conditions, such as non-arable land and brackish water. Here in New Mexico, we have some huge saltwater aquifers that would be ideal for algae growth because nothing else can really live in them. Algae can also be harvested more often than annual crops like corn and soy, and it produces more oil per acre than plant-based biofuels. There are some challenges, however, in producing algae for biofuels. One of the challenges is that we need it to have higher productivity and accumulate the biomass faster. And we want to increase the carbon storage because this is what we will be using to create the biofuels. We need it to grow well in brackish waters and a variety of salt conditions. And the reason why that we need it to grow in a variable conditions is because we have outdoor ponds where the algae is grown, and the salinity of those ponds varies considerably depending on rain and evaporation. And another benefit to growing it in salt water is that it's more sustainable because, as you can see here in this picture, the huge bulk of the world's water is salt water or brackish water. And so to grow the algae in salt water, it wouldn't compete with fresh water that we need for our consumption. So to test how this genus of chlorella does in different salt conditions, we did a salinity tolerance test, and our goal was to see how these different strains grew in different levels of salt concentrations. So the reason we chose chlorella to study is because it's a freshwater algae that has been very well studied in the lab, that grows fast, and it produces in a moderate amount of lipids and carbohydrates, which is what we're going to use to create biofuels, and it grows fairly well in brackish water. So we tested 4 different strains in 6 different salinity concentrations, and every day we took samples and measured the biomass productivity with optical density measurements. So as you can see, these are the graphs of the 4 different strains. We had 2 strains from the Sorokiniana species, the 1228 and LANL strain, and we had 2 strains from the Desiccata species, which was the 2437 strain and the 2526 strain. And you can see here in the graphs that the 1228 did very poorly at higher salt concentrations. And the other three performed better. The Lannel strain was not fabulous, but it did grow in all of the concentrations of salt. But the Desiccata did really well. It grew in all concentrations and it grew well, better than the Lannel strain. So this set of graphs is the same information, but we compared the strains and we grouped them by salinity. So at the top are the lower amounts of salt, and you can see that every strain did well. And on the bottom is the higher concentrations, and there's it's very clear that the two Desiccata strains did really well at the higher salt concentrations compared to the other two. And the Lannel strain did grow some, but not great. So the conclusions of our salt tolerance screening, we had studied the Chlorella sorokiniana 1228 strain a lot in the past, so we did not expect it to grow at the higher salt concentrations, and like we expected, it did not. The higher concentrations of salt samples were clear and the cultures crashed. The Lannel strain did well, and it does have potential for further study. It grew at all salt concentrations. However, at the highest concentration, it did not ever starve of nitrogen, which implies that it didn't grow well. The two Desiccata strains were the best species in our study due to their rapid growth and tolerance of all salt levels. The samples were very green, and once they starved of nitrogen rapidly, the cultures turned yellow, which is very indicative of lipid accumulation. So these two strains have a lot of potential in our further work. For our future directions, we want to run the experiment again at higher volumes to create samples for further analysis, such as transcriptomics, where we'll study the changes in gene expression at the different salt levels. We also want to run a lot of biochemical analysis to determine what types and how much lipid was accumulated and how much carbohydrates was accumulated, and to possibly study the protein content as well. We also want to study the ash-free dry weight, which is a measurement of the total biomass that was produced. I would like to thank Lannel and Beto for providing us with funding for our research, and Dr. Terica Dale for being a great leader and mentor, and Claire Sanders for her excellent mentorship and helping me with every step along the way, and Nalusha for giving us background info and doing a lot of the work that helped with protocols leading up to what I was going to do, and Zeph for protocols and also excellent spreadsheets. So thank you.